Okay, welcome to your lecture on the human microbiome. Now on to our first slide. Okay, the microbiome is the total of all of the microorganisms, including bacteria, viruses, and single-celled eukaryotes like protozoa and fungi, living in association with the human body. The Human Microbiome Project was launched by the National Institutes of Health in 2007 with the mission to generate the resources and expertise needed to characterize the human microbiome, and also to check and see how it related to health and also disease. What they found was that there is more diversity than we ever thought possible living within and on the human, right? Living in and within your body. With that project, if you take a look at the picture right here, it shows the multiple sites that were sampled. So they actually took volunteers throughout all over, and they had to sample different parts of their body and send it in, and then they did genetic sequencing to figure out which types of organisms were living where in the body. Very interesting. So if you take a look at the other picture, okay, well, on the far left, the one with the graph, what it's showing you is that about half gave us we know about half that was living there, we were able to characterize. We knew something about them. And the other half, we don't really have a whole lot. So we had undetected pieces, parts and pieces. We had some that the sequences really didn't match with anything else that we know about. And about 40% didn't match in our database. Okay, so about half that we found living in and on the human body, we could characterize. The other half we couldn't, and that's a big chunk of them. So lots to learn from our human microbiome. When we study the human microbiome, it's really a lesson on ecology. It's learning about the habitats and interaction between the microbes and the human host, also between the microbes themselves. Now, kind of what we call it. The human microbiome, when we're talking about the organisms themselves, we usually refer to it as the microbiota, or an older term for it, the normal flora. These microbes usually colonize us without normally causing disease. So if they kind of stay put where they're, supposed to, they don't usually cause us problems. Once the microbiota was thought to be commensal, a type of relationship, type of symbiotic relationship, and where one benefits and one does not benefit. It's neither harmed or benefits. But now that we know more about the microbiota, that we think that most are what are called mutualists, another type of symbiotic relationship where both benefit. So both the microbe benefits and the human host will benefit. We're learning more and more about how the microbiota, those resident bacteria and other types of microbes impact our overall health. Links and connections between the microbiota and other parts of our body, and either how our body functions, have been uncovered in recent research. So look forward in the next couple years for us to learn a lot more about that with some up and coming research. Now, where are they found? I said that most is found within the GI tract of the body, and that's even starting with the mouth. So you want to think mucous membranes, anything that's coming into contact with the air. So the skin, the mucous membranes, that will include our entire digestive tract and also urogenital, I should say. If I had to pick two places where we're going to find the most, it's going to be in the skin and the large intestine is where we find the most there growing. There's a video I'd like you to watch. There's a link at the top of this slide. So right up there, watch that video, watch that short video on Meet Your Microbe. microbiome, and I will see you on the next slide. Now I want to talk about the beneficial or protective role of the normal microbiota, or the microbiota. And I'm going to list out 4 for you, and we're going to start with number 1, which is competitive exclusion of pathogens. So if you take a look at the picture on the far left, where you see some green things and some red things, you can blow that up and kind of take a look at that. And what that's representing is the gut wall. So let's think about this. The microbes in the small intestine, in the large intestine, in the GI tract, we'll just say GI tract, they live along the walls. Okay, so they're attached to and they live along the walls of those cells. And as long as we have the microbiota there, kind of our normal flora there, what does that do is it's covering up some binding sites that would otherwise be available to pathogens. So as long as we have some good beneficial bacteria there attached to the walls, we're kind of limiting the space for attachment for the pathogens. The picture on the right, it's a little bit more blown up. You can see that a little bit better. And the green is representing the beneficial bacteria, the normal residents, the microbiota that we would typically find living in the GI tract. And so if they're taking up all of the space, there's not really a whole lot of wiggle room for those pathogens to attach and therefore exert their negative effects on those cells. So this is one of the ways in which you have competitive exclusion of those pathogens. It's not leaving space enough for those pathogens to attach. Another example of competitive exclusion of pathogens is that if the normal microbiota is there, normal flora is there, it is going to be consuming most of the available nutrients and keeping it away from the pathogens. So leaving very little for the pathogens in order to metabolize and multiply there. So if all of the resources are gone, then it doesn't leave a whole lot for the pathogens. So that's another example of competitive exclusion of the pathogen. Okay, now we're on to talk about another way that our microbiota play a beneficial role or a protective role, and that is by producing toxins or antimicrobial substances. So let me explain that one a little bit. The microbiota produce toxic molecules that would prevent the growth of other microbes and potentially pathogens. So let me give you some examples. There is one type of bacteria known as Propionibacterium. And notice I have the SP after it. That means species of Propionibacterium. They live on the surface of the skin, and it degrades lipids. So it degrades lipids, releasing what are known as fatty acids. The fatty acids then inhibit the growth of some pathogens. Kind of cool. Another example is one that's known as Lactobacillus. So species of Lactobacillus, and we find these all over our body, and one of the places is in the vagina. So it produces lactic acid. Lactobacillus, that kind of makes sense now, produces lactic acid. And the acidic conditions, because of the lactic acid or the low pH, prevents the growth of some pathogens. Nice. Okay, a third example is a species of Bifidobacterium. And the Bifidobacterium produce bacteriocin, which is an antibacterial property compound. This compound helps prevent the growth of Listeria monocytogenes. So there's the bacteria that are producing these toxins, these antimicrobial substances, And those antimicrobial substances aren't having an effect on us, it's having an effect on these other types of microbes, and it's preventing the growth of some that could be pathogenic. Pathogenic are the ones that make us sick. Okay, another way that our microbiome plays a protective role, benefits us, is that it helps to train or stimulates parts of our immune system. So let me explain that one a little bit. The body's immune cells can learn the difference between harmless and pathogenic organisms. So if we have harmless organisms, or what we call friendly organisms, living in and on us, then our immune system can learn the difference between what good and bad. Another example is by stimulating the production of IgA. So if you remember what IgA is, it's a type of antibody, and it's a class of antibody, and it is found in mucous membranes. So our microbiota is stimulating the production of IgA in our mucous membranes, and that IgA can then bind to the pathogens. Okay, so another way it benefits is by providing beneficial molecules to the host. And this is carried out by the bacteria and yeast. Yeast is a type of fungi, but mainly bacteria. Types of bacteria that have this ability are the Bacteroides, species of Bacteroides, species of Lactobacillus, species of Bifidobacterium, some types of E. coli, and in general, the members of the phylum Proteobacteria. All provide types of vitamins and various vitamins. So to give you some examples, vitamin B, which is important for glucose metabolism, folic acid for developing cells. Bacteria produce folic acid, we do not. And so we get a lot of our folic acid from the bacteria living in our intestines. Vitamin K, which is important for blood clotting. And the bacteria will metabolize the food, the indigestible food in the GI tract, indigestible by us, and then they will metabolize that and secrete vitamin K or the other vitamins or the other substances, and then we will absorb them. It's kind of cool. Okay, what other types of molecules do they secrete? Are short chain fatty acids. And the short chain fatty acids are as a result of that bacterial metabolism. And then those short chain fatty acids are absorbed or used by the epithelial cells that line the GI tract. What does it do? It helps to reduce inflammation in those areas. So we're benefiting from those molecules that have been secreted by the bacteria mainly. Okay, building on the last slide, what we talked about, them secreting substances that we can then absorb and use to our benefit. You've probably heard of probiotics. So probiotics that is a marketing term, and it is originally it was a marketing term, and it's live bacteria and types of yeast and possibly some other types of organisms that offer a health benefit. So we can take them as a supplement. So if you've ever taken probiotics, you can have dehydrated. They come in pill form. We can also find them in various dairy products like yogurt. Up on the screen, I have the supplement facts, kind of the back to some dehydrated bacteria that we take in supplement form, in pill form. And what probiotics in general are live microorganisms that offer a health benefit. We want these to take up residence in our body. So why would we eat yogurt? Why would we take probiotics? Is that we want them to take up this residence. We want to take it to the stomach, survive the conditions of the stomach, make it to the small intestine, mainly make it to the large intestine and set up shop there. We're kind of seeding our large intestine with bacteria that we know is beneficial. So lots of different types of probiotics. There used to be only one type of probiotic historically, and it was lactobacillus. We were really good at culturing lactobacillus acidophilus and using that as our probiotic in yogurt and other types. Now, the more that we know about the different strains, the different species, we've started to include lots of different types in there. And especially when we start getting the links between our normal microbiome, the microbiota, and the health links, we'll probably see that probiotic, those strains start to change as well, what we're including in them. You may hear another term kind of associated with probiotics, and that's called prebiotics. And what is a prebiotic? It's not a microorganism at all, but it refers to the food that we eat that is metabolized by our intestinal bacteria. And it's usually like the high fiber foods. The high fiber foods that can't be digested by us, but can be digested by bacteria. So it's kind of, yes, we want to seed our intestines with this good bacteria, but then we also want to give it what it needs in order to metabolize to make these compounds that we want to then absorb. So I have a short video for you to watch linked on this screen. So just click right here, prebiotics and probiotics, a short video for you to watch on that, and then I will see you on the next slide. Okay, now I want to talk about the harmful role of our microbiota. How can it be harmful to us? And one of the ways it can be is if it's introduced to otherwise sterile sites. This can lead to opportunistic infections. Microbes may become harmful if they're introduced to other sites of the body where they're not normally found. So for example, the first example I'm going to give you is it has to do with skin bacteria and placing an IV. So on every surface of our skin, we're going to find our normal flora, our microbiota, and a lot of it is bacteria. So we have our typical skin bacteria, and I'm going to give you an example of Staphylococcus epidermidis. This is one that you will learn about in lab, and it lives just fine on the surface of our skin and gives us that protective role as well as you just learned about. But it could be introduced to the bloodstream. Let's say you're placing an IV and you're poking through the skin where the bacteria is, so you could basically deposit it into the bloodstream. If it gets to the bloodstream, it can cause an infection. Okay, so what do we do to prevent that? If you've ever placed an IV or had an IV placed or even had a shot, what do they do? Yeah, you probably got it right. All right, they rub something on you. They rub an alcohol swab. So they take the alcohol swab, rub it on the surface of your skin. And why are they doing that? And that is to get rid of the bacteria that's going to be in that area where they're going to pierce the skin. Now that alcohol swab, it needs to that alcohol needs to come into contact with the skin for about 30 seconds in order to kill that bacteria that's there. So it doesn't get penetrated into the skin, into those dermal blood vessels or into the blood vessels at all, and potentially cause an infection. All right, so another example is E. coli. E. coli is a normal inhabitant of any mammal's intestines. We are mammals, and so humans. It's a normal inhabitant. I would expect to find lots of E. coli in our intestines. However, now while it's there, it gives us some vitamins. We'll absorb those vitamins. However, if the E. coli is introduced into other parts of the body where it's not normally found, like the urinary tract, for example, can lead to a urinary tract infection. So if by chance that E. coli from the feces or the large intestines get deposited very close to the urethral opening, it can potentially lead to a urinary tract infection. That same E. coli, if introduced to our eyes, our nose, our mouth, it can also lead to an infection. Okay, another way it can be harmful to the human host is suppression, and this, you gotta hang with me on this one, is suppression of large groups of the normal microbiota or normal flora. Antibiotic treatment kills the majority of our bacterial microbiome while it's trying to fight off the infection caused by the pathogen. So when we're taking antibiotics, yes, we want it to kill the pathogen, but as it turns out, it will also take out a lot of our microbiome, okay, giving us that protective role. So if we're taking out large portions of our microbiome, that's what I'm talking about. So let me give you some examples here. In the vagina, we talked a little bit earlier about species of lactobacillus. And if lactobacillus is now gone, physically gone, this now frees up space. So the tenants are now gone, and it frees up the space for overgrowth of other types of organisms. Now, if you're taking antibiotics, that's geared to kill bacteria. And there's other types of microorganisms that make up our microbiome, our microbiota. One example is a yeast known as Candida, Candida albicans. There's other types of species, but this is the species we're going to use today, Candida albicans. These are yeast cells, so if we kill off large portion of our microbiome as far as bacteria goes, we have a lot of free open space so that Candida can now grow. And that's what it does. It grows and it overgrows. And the overgrowth of this Candida can lead to problems. The overgrowth in the vagina of Candida, species of Candida, can lead to what we call vulvovaginal candidiasis. And you may not know it by that name, but it's a yeast infection. And this can happen in the mouth as well. You can take a look at the pictures and you can physically see it, right? You can see that overgrowth of the yeast. It's kind of a whitish to a off-white color, kind of soft and fluffy, and that is the Candida growing there. So it can happen in the vagina. It can happen in the mouth. You can have that overgrowth in the mouth. We call that thrush, but it's from the overgrowth. Now you might be red and itchy and painful. It's usually maybe like a bright red color and swollen. Any of those areas that we have this overgrowth of the Candida. And really that red, itchy painfulness is coming from our immune system. Our immune system starts to work and gets a little freaked out, and we get a lot of swelling and redness, inflammation. You're familiar with that. And it's the reaction over stimulation of the histamines as well. And so our immune system is alerted because of the overgrowth of the Candida. And so then we have these signs and symptoms of that particular type of overgrowth or infection. Okay, I'm going to give you another example with, suppression of large groups of microbiota. So, that may have happened to you before where you were taking some antibiotics and you either resulted in maybe a yeast infection or a or thrush. And by the way, guys can get yeast infections as well. This can be not only just in the vagina, but it can be in the urethra as well. So, but I want to give you another example. And this example happens a lot of times when people are consistently on antibiotics. So for whatever reason, they are consistently getting bacterial infections one after another, hard to treat infections. So they are receiving antibiotic treatment and then maybe another one very quickly. We see this a lot in elderly individuals when they are more susceptible to bacterial infections. So that's what I mean by consistent antibiotic use. So consistent antibiotic use can also inhibit the growth of intestinal bacteria and allow for the growth of highly antibiotic resistant species to grow. Okay, so we're wiping out the majority of them that are there and the bacteria that is there in the intestines, but they happen to have these highly resistant strains, resistant to the antibiotics. And there's one that I want to talk about, you've heard me talk about before, and that is called C. diff. The slang term for it is C. diff. And that one type is this highly resistant species known as Clostridoides difficile. Now, that's a new name. There was a name change in 2016 from Clostridium difficile. So when I learned it, it was Clostridium difficile. 2016, they changed the name to Clostridoides difficile. The more that we've learned about it, the more genetic testing we've been able to do on it. And the taxonomists say, nah, it really needs to be in this other group. And so they changed that group. They still have Clostridium, but now the organism that was once known as Clostridium difficile is now Clostridoides difficile. Didn't lose the C, so we can still give it its nickname of C. diff. Now, C. diff, it can produce toxins. So some strains can produce toxins that cause diarrhea and colitis. Not a good feeling, okay? It's a gram-positive rod that produces endospores. We do know that the number of outbreaks of this particular organism is increasing. Some patients can acquire it in the hospital, while other patients carry it as a minor component of their microbiome, as their microbiota. And so it does pose a major problem because when you're trying to fight infections from pathogens, we understand that we're going to wipe out our good bacteria in the large intestines, but we're going to come across some that are resistant, like this individual, this type of species. And we can really have some harmful effects from this species. So I have a short video for you to watch that shows about the C. diff. Okay, so watch that short video, and then that will be the end of our talk on the human microbiome.